Hello everyone, this is TIMS voice, and today we're going to talk about the first pathology lecture in the CNS module using TIMS paper. The first pathology lecture is titled The Neural Response to Injury. The Neural Response to Injury. So we're going to talk about the reaction of the cells in the CNS after any injury. As we all know, the CNS does not only contain neurons, it contains different types of cells, like the glial cells. So we're going to divide our lecture today into three divisions. The first two parts, we're going to talk about the reactions to that injury on the cell level. So we're going to talk about the neurons first, and then the glial cells reaction. At the third part, we're going to take a step back to look at the whole tissue and talk about the circulatory changes and edema. The circulatory changes and edema. So first of all, we're going to talk about the neurons. The neurons are composed of the cell body and the axon. So we're going to divide this part as well into two parts. First of all, the neuronal response. The neuronal injury is caused by hypoxia, which means lack of oxygen, ischemia, which is a consequence of hypoxia, and other insults. These other insults include inclusions, which we will be talking about later in this lecture. Other insults also may include any hypoglycemic insult. Any of these insults or any of these damages or diseases may affect and harm and injure the cells of the central nervous system. So these injuries may lead to, first of all, cell shrinkage. Of course, we're here talking about the cell body of the neuron, not the whole neuron. Okay, so first of all, cell shrinkage and nuclear pyknosis. So the cell body becomes smaller and the nucleus undergoes pyknosis. So let's recall together the definition of pyknosis. Pyknosis means the irreversible condensation of chromatin inside the nucleus when the cell undergoes apoptosis or necrosis. So basically, when the cell is dying, the cell shrinks and the nucleus undergoes pyknosis. It's the irreversible condensation of chromatin inside the nucleus. So as we can see in this very first picture, the nerve cells are a little bit smaller than usual, and you can compare it to the normal microscopic picture of neurons, as well as the nucleus. The nucleus is a little bit darker as a consequence of pyknosis, and it's a little bit smaller too than the usual. Okay, so first of all, the cell is smaller because it shrank, and the nucleus is a little bit darker and smaller as well. So this was the first reaction. The second reaction is loss of Nissl substance and central chromatolysis. First of all, we need to know what is the meaning of Nissl substance. This Nissl substance is a part of the rough endoplasmic reticulum of the neuron. Okay, it is composed of it's a network that exists around the rough endoplasmic reticulum. It's a part of the rough endoplasmic reticulum. It may also contain polyribosomes, rosette-shaped polyribosomes. Okay, and central chromatolysis. It means that the nucleus as well as the other contents of the neuron are going to be distributed at the peripheral side of the cell, as we can see in this second microscopic picture. Okay, so we have these neurons. First of all, the last one, this last cell. Okay, as you can see, there is absolutely nothing in the middle of the cell. It's so clear, and the contents of this neuron were distributed to the peripheral side of the cell. This is what we call central chromatolysis, and there is no cell substance as the other cells. Okay. So this was the second response or the second reaction. Number 3, intense cytoplasmic eosinophilia or red neurons. The last microscopic picture reveals the abnormal color of these neurons using H&E stain, of course. The neurons here are more red or more a little bit more pink than in color than the other neurons, and you can compare this third picture to the previous two. The previous two were a little bit at the early stages of this response, so they are not so red as the third one. Okay, so that was the third response. So again, the neuronal injury is caused by hypoxia, ischemia, or other insults. This may lead to cell shrinkage, nuclear pycnosis, loss of cell substance, central chromatolysis, and intense cytoplasmic eosinophilia or red neurons. So the neuronal injury becomes clear using the H&E stain, which is obvious considering the colors. Now we're going to talk about the axonal response. Injured axons undergo swelling, forming spheroids, and so disruption of transport. As we all know, the function of the axon in the central nervous system or in the neuron itself is to transport signals. From one cell to the cell body of the same cell. Okay? So when the axon is injured, it undergoes swelling. So it swells, it becomes a little bit larger, and then it coils around itself, forming spheres. These spheres is what we call axonal spheroids, and show disruption of transport. So the axon is no longer able or capable of transporting these signals. So it first of all swells, and then coils around itself, forming spheres that look like a circle, a big circle in the under the microscope. Okay, these circles or these spheres are called axonal spheroids. Okay, so this microscopic picture is taken using the H&E stain. Okay, there are other stains, of course, that we can use to observe these axonal spheroids, including the silver stain in this second picture. As you can see, this axonal spheroid pointed at by this arrow is a little bit more clear than the previous one. We can also use any other stain like the immunohistochemistry stain. Okay, so this was the axonal response. Neurons are the most vulnerable cells to injury of the CNS, as they don't have energy storage and have high functionality. What does that mean? Okay, so normally any action, any voluntary or involuntary action that or any physiological processes that occur inside your body are going to be controlled by the neurons. Okay, so neurons in your central nervous system work all the time. They don't stop. There is no rest. So their demand of energy is so high, and whatever energy is produced by the powerhouse of the cell, the mitochondria, is immediately used so that the neuron can live. So it consumes a lot of energy, almost all the energy available in the cell. So there is no storages. There is no more energy than what the neuron needs so that it can store it. There is no storages at all. So whenever any neuron is injured, there is no energy stores to start the process of regeneration or to fix the neuron. There is no energy enough for the neuron to live. And just imagine if the powerhouse of the cell, the mitochondria itself, was damaged or was injured. There is no stores, that the neuron will immediately die. So the neurons, that's why the neurons are the most vulnerable cells to injury. Okay? Number 2, axonal injury can lead to neuronal injury and vice versa. When the axon is injured, this can lead to the injury can move to the cell body as a consequence to the axonal injury, and vice versa. Both can, if one of them is injured, the other one will be injured as well. Okay. Now we're done with the neurons. We're going to talk about the glial cells reaction. The glial cells are the astrocytes, the oligodendroglia, and the ependymal cells. Each of them has a very important function in the central nervous system, because they all work as a supportive tissue to these neurons. So as we said, the astrocytes are the principal cells responsible for repair and scar formation in the brain. Okay, so they're basically the fibroblasts of the central nervous system, except that they are not fibroblasts, of course. They are astrocytes. They are responsible for repair and scar formation in the brain. So after any injury, how can these cells respond? The astrocytes undergo hypertrophy, which means they enlarge in size, okay, and hyperplasia, they increase in number as well. So they enlarge in size and increase in number, both hypertrophy and hyperplasia. The nucleus enlarges as well and becomes vesicle-like nucleus, okay. So the nucleus of the astrocyte after an injury becomes vesicle-like. It enlarges, the cell itself enlarges, and the cell undergoes hyperplasia as well. The cytoplasm, of course, when the cell enlarges in size, the cytoplasm will expand as well and takes on a bright pink color, as we can see in the first picture here. The nucleus is a little bit vesicular. The cytoplasm is a little bit brighter than the surrounding tissue or the surrounding cells, and the cytoplasm and the cell itself are larger than normal. Okay. All right, and the cell becomes plump, larger. Plump means a little bit larger, and so called gamestocytic astrocyte. Gamestocytic astrocyte is what we call an astrocyte after the morphologic changes as a consequence of any injury. Okay, we call it gamestocytic astrocyte because of its color, because of its size. They say it looks like the RBCs, which is why it's called gamestocytic astrocyte. Number 2, in long-standing gliosis, and by gliosis we mean the astrocytic reaction to injury. This whole morphologic changes and reaction is called gliosis, when talking about the astrocytes, of course. So the cytoplasm of astrocytes shrinks in size, and the cellular processes becomes more tightly, and we call them fibrillary astrocytes. So the cytoplasm of an astrocyte in long-standing gliosis. So first of all, in the early gliosis, okay, as we said in the first point, it undergoes hypertrophy, hyperplasia, the nucleus enlarges, and the cytoplasm expands. Basically, the cell becomes larger. But in long-standing gliosis, later on, okay, the cytoplasm of astrocytes shrinks in size. It becomes smaller, okay, and the cellular processes, the astrocytes have cellular processes. You will know that if you have studied the histology, the first histology lecture, okay. So the cytoplasm becomes smaller, Okay, and the cellular processes becomes more tightly. We call them fibrillary astrocytes. As we can see in this second picture, the astrocytes are smaller than usual, okay, and the processes are a little bit tight. So we call them fibrillary astrocytes. Number 3, corpora amylacia. The corpora amylacia are starch-like bodies, okay? They are some kind of inclusions. Somehow, they are lamellated polyglucosan, okay? So they are made of carbohydrates, and they increase in number with advancing age and represent a degenerative change, okay? So corpora amylacia are carbohydrates. We call them starch-like bodies, okay, which are lamellated polyglucosan bodies. They increase in number with age and represent a degenerative change, so they harm the brain. Number 4, you can see this in the microscopic picture number 3. Those are the corpora amylacia or starch-like bodies. Okay, so number 4, we're going to talk about the rosenthal fibers. Okay, the rosenthal fibers, these fibers over here, yeah, these fibers are thick, they are elongated, they are brightly eosinophilic protein aggregates. So the corpora amylacia was carbohydrates, the rosenthal fibers are protein aggregates, okay, found in astrocytic processes in chronic gliosis, in chronic at the late stages of gliosis. We can see the rosenthal fibers, which are the protein aggregates, in the astrocytic processes in chronic gliosis and in some low-grade gliomas. Now these gliomas, the low-grade gliomas, are tumors that are, it is formed, these tumors are formed of the oligodendroglia and the astrocyte cells, okay? So this harms the brain as well, or the central nervous system. So whenever you have a low-grade glioma or chronic gliosis, at these stages, you can observe the rosenthal fibers in the astrocytic processes, okay? And the corpora amylacia are starch-like bodies. They are lamellated polyglucosan bodies that can be observed in any degenerative change in the brain or the central nervous system in general, okay? So other glial cells response. We have different glial cells other than the astrocytes. We have the oligodendroglia, we have the ependymal cells. The oligodendroglia, why isn't it so important as the astrocytes? Because the oligodendroglia exhibit a limited spectrum of specific morphologic changes. They don't change completely like the astrocytes. We can't observe the change very clear because it is so limited. There is no many changes as the astrocytes. So the oligodendroglia exhibit a limited spectrum of specific morphologic changes in response to various injuries. Oligodendroglia apoptosis is a feature of demyelinating disorders. So what is the oligodendroglia? In general, the oligodendroglia are the Schwann cells that make the myelin sheath in around the neuron, around the axon of the neuron. Except that the oligodendroglia exists only in the central nervous system, while the actual Schwann cells exist at the peripheral nervous system. Okay, so whenever you see oligodendroglia apoptosis, when this cell is undergoing self-suicide or apoptosis, okay, this is a feature or a change that can lead to realizing that this patient have demyelinating disorders, okay, because this oligodendroglia are responsible for the myelin, for making the myelin sheath in the central nervous system. So whenever you can observe them killing themselves, undergoing apoptosis, this is a feature of demyelinating disorders. And then we have the ependymal cells. The ependymal cells do not regenerate. The ependymal cells do not regenerate. They form ependymal granulation if exposed to injury. So the response of the ependymal cells comes in the form of ependymal granulations. Okay, all right, and then we have the choroid plexus. So what is the choroid plexus? It's a network of blood vessels in each ventricle of the brain. As we know, the brain has many ventricles, and it is responsible for the secretion of cerebrospinal fluid. It is responsible for the secretion of the cerebrospinal fluid, CSF, of course. So how are the ependymal cells related to the choroid plexus? The ependymal cells contribute to the secretion of the cerebrospinal fluid as well. Moving swiftly on to the last type of glial cells, the microglial cells response. The microglial cells are basically, they do the same function as the macrophages in our bodies, okay? So following injury, it can respond in different ways. First of all, microglia proliferate and develop elongated nuclei, and then we call it rod cells. So they are rod-shaped cells, a little bit elongated, as we can see in this first microscopic picture. Okay, the microglial cell is elongated. It's more elongated than normal, with an elongated nuclei, and that's how we know that this tissue has been injured. Number 2, and/or aggregate around dying neurons. So basically, the microglial cells can either do or either respond in one of these ways, or two of them, or all of them. Okay, so it aggregate. So it may proliferate, develop elongated nuclei only. Okay, and it can do that, and as well as aggregating around the dying neurons. Okay, as we can see in the second picture, these darkly shaped, darkly colored cells aggregate around this pink neuron as it dies, so it can function and start the neuronophagia process. Neuronophagia, okay, the phagocytosis of a neuron. Okay, number 3, it can form aggregates around necrotic foci. So the microglial cells, okay, after any viral infection that leads to any dying neurons, okay, the microglial cells aggregate around the necrosis, the part where the necrosis occurs. As you can see here, can you see the cells, the microglial cells? They aggregate, okay, to form microglial nodules. So when you see this third picture, you know that there is a viral infection somewhere, such as HIV. This note here says that oligodendroglial cells are the most susceptible among glial cells, followed by astrocytes, while microglial cells and vascular endothelium survive the longest. So the neuron is the very first, or is the most susceptible among all of the central nervous system cells. Okay, but speaking about the glial cells only, then the most susceptible cell is going to be the oligodendroglial cell, and then the astrocyte, and then the microglial cells and endothelium. because the previous two can survive the longest. Okay? Now let's move to, we're now taking the step back to look at the whole tissue and watch the changes that may occur in this tissue after an injury. So we're going to talk about the cerebral edema. So whenever there is a fluid trapped around the brain, okay, more than the usual fluid, this is what we call edema. So edema is an accumulation of excess fluid within the brain parenchyma. Okay? We have two types of edema that often occur together. The first one is physiogenic edema. The second one is cytotoxic edema. So physiogenic edema occurs when the fluid shifts from the vascular compartment to the extracellular spaces of the brain. It can either be localized or generalized. So basically, we have blood vessels going in the brain. Okay, so physiogenic edema occurs when the fluid compartment of the blood vessels themselves move from these vessels outside to the extracellular compartment between the neurons. Okay, so at this type, physiogenic edema, the issue is usually in the, it starts from the blood vessels, so that the fluid can move from inside these blood vessels to the extracellular compartment of the brain, causing a swelling. So it can be localized as an increased vascular permeability due to inflammation or tumors. So if we have an inflammation in a certain area of the brain, if we have a tumor in a certain area of the brain, physiogenic edema is going to be a consequence to that injury or tumor, okay? And this is when it's localized. Or it can be generalized in the whole brain or sometimes in the whole body. So this was the physiogenic edema. Number 2, the cytotoxic edema is an increase. As we know, cyto means cell, toxic, it's almost the same meaning as death. So it's an increase in the intracellular fluid, and it's secondary to neuronal and glial cell injury. So the neuron is one of the cells that after is severely injured, okay, it has a cytotoxic activity. So it kills itself in a process we call apoptosis. Okay, so cytotoxic edema is a part of the apoptosis of the neuron or the glial cells after any injury. So cytotoxic edema is increased in the intracellular fluid inside the neuron itself. Okay, and the issue here is not in the blood vessels, not at all. No, the issue is in the cell membrane, in the cell itself. So the cell, after any severe injury, is going to kill itself by apoptosis, and one of the mechanisms is cytotoxic edema. The membrane of the cell is ruptured, and the extracellular fluids between the neurons enters the cell to cause it to die. Okay, as might follow generalized hypoxic or ischemic insult or exposure to certain toxins. Okay, so cytotoxic edema and physiogenic edema almost occur together at the same time sometimes. So if we have an inflammation in a certain area of the brain, the vascular permeability is going to be increased, so we're going to have physiogenic edema, and at the same time the inflammation is going to be caused by, the inflammation itself is going to be caused by any infection, so neurons are dying, so apoptosis is occurring, so we have cytotoxic edema as well. What are the complications of edema? First of all, it's going to raise the pressure inside the fixed capacity of the skull. So we have the skull is a hard bone, okay, that is containing the brain in a certain space. Okay, it have the ability to contain and cover a certain space. Okay, so whenever the space is getting larger or expanding by the fluids of the edema, okay, this is going to cause the pressure inside the skull to be raised and increase the intracranial pressure. Number 2, herniation. Herniation is a protrusion, as we can see here. If you can see this arrow, this part of the brain was not supposed to protrude like that. It was supposed to be preserved and fixed in its place. Okay. However, after the edema, the cerebral edema, it caused it to herniate. Okay. So this is the herniation. Number 3, it may cause infarction. So the complications are increased intracranial pressure. Number 2, herniation. Number 3, infarction. So today this is. Remember when we talked up there about the insults that may cause the neuronal injury? As we said, it can be caused by hypoxia, ischemia, or other insults. And I said that these other insults include the inclusions. Okay, so what are the inclusions? The inclusions are substances that are different in the histological structure from the CNS and got included into the cell. So the normal histology of a neuron does not contain these bodies, does not contain these inclusions. Okay, but if they pop in this cell, or if they were deposited in the cell, they may cause an insult, they may cause a neuronal injury. So we have the intracellular inclusions. In some neurodegenerative diseases, we have these inclusions may cause some neurodegenerative diseases. For example, the Lewy bodies in Parkinson's disease, okay, and tangles in Alzheimer's disease. Number 2, these inclusions may be pathogenic viruses. Sometimes they may be pathogenic viruses. Number 3, accumulation of lipofuscin in the cytoplasm and lysosome with age. So with aging, okay, accumulation of lipofuscin occurs and it may harm the cell. So we're going to continue to the appendix, which are some really important information the doctor recommended that we must understand. First of all, the average weight of the brain is 1,400 grams in men and 1,250 grams in women. Number 2, the brain does not have lymphatic drainage, which is important when we move to the chapter talking about tumors in the brain. Okay, the brain has no energy stores of its own except for a small amount of glycogen in astrocytes. So we said that earlier. We said it's why the neurons are the most vulnerable cells. This is the more susceptible cells to be injured, because they have no energy stores of its own except for a small amount of glycogen in astrocytes. So whenever the neuron is injured and needs energy immediately, it uses the energy in the astrocytes, but it won't be enough until just few minutes. It will be enough for the neuron for only few minutes before it dies. And then the brain is 200 of the body weight is in the brain only. However, it takes 20% of the cardiac output and 15% of oxygen consumption, which increases the risk of hypoxia. Okay. So the gray matter is more vascular than the white matter, as it has a greater metabolic requirements. The gray matter contains the bodies of the neuron as well as the blood vessels. It's more vascular, okay, because of the body of the neuron have a greater metabolic requirements than the axons, which exist in the white matter, okay. That's why the gray matter is more vascular. And then there may also be variations in local blood flow in relationship to metabolic demands. So the more this area of the brain have a high metabolic demand, okay, the more it had a local blood flow. Energy depletion causes failure of, okay. So remember when we said that when we talked about the neuronal injury, remember when we said that the neurons cannot deal with any energy depletion? That's because energy depletion causes failure of the sodium-potassium pump. So now what will happen? What will happen is that the synaptic function and conductivity will cease at this point, okay? And the membrane potential will still be depolarization, will stop and cease at this point, okay? Then what will ruin everything now is that depolarization is going to cause the neurons to release the neurotransmitter called glutamate into the synaptic cleft, okay? So what does the glutamate do? The glutamate in excessive amounts, such as in this cases, is an excitotoxin, okay? It causes this is how the neuron undergoes apoptosis, okay, by releasing excessive amounts of glutamate, and glutamate is an excitotoxin to kill itself. It causes a passive influx of chloride and sodium into the cell. The glutamate causes passive influx. It needs no energy, without any need of any energy. It causes a passive influx of chloride and sodium into cell, causing osmotic or cytotoxic edema and rapid death. So because a lot of chloride and sodium, a lot of ions are moving from the synaptic cleft into the cell, okay, the water or the fluid in the extracellular compartment is going to follow these ions inside the cell, okay, causing cytotoxic edema and rapid death. Additional structural damages, we will not only stop here, but additional structural damage develops hours or days later as a result of calcium influx into neurons. So we will not just have chloride and sodium influx, we will also have calcium influx following the injury, after hours to days after the injury, will cause a structural damage. How is that? Calcium activates the catabolic enzymes which exist inside the cell. These catabolic enzymes are proteases, phospholipases, endonucleases, okay, as well as activating nitrogen oxide synthase. Nitrogen oxide synthase is an enzyme that synthesizes the nitrogen oxide, okay, which is a free radical. So this free radical nitrogen oxide, as well as the catabolic enzymes, the proteases, the phospholipases, and the endonucleases, they destroy the cellular contents, causing more damage. So we have the cytotoxic edema, and then destroying the cellular contents using these catabolic enzymes and the free radical nitrogen oxide. Neurons are more sensitive than glial cells because they have higher energy demand, as we said, and only they produce glutamate, okay? So what starts all of this process is the glutamate. Some neurons are more vulnerable than others as they are bigger in size, which requires higher energy. Okay, remember when we said that the neurons are the most vulnerable cells in the central nervous system. Yes, there are variations between neurons. Okay, so bigger neurons need more energy. So with any slight depletion or decrease in energy, they are going to be more vulnerable, okay, to get injured. And yeah, these the big neurons exist in few locations inside the brain, like the hippocampal pyramidal cells, CA1, pyramidal neocortical neurons in the layers 3, 5, and 6, Purkinje cells, and striatal neurons. They all have the highest vulnerability to get injured. So the human brain will appear normal on autopsy after immediate death. So if the human brain was injured, or these neurons were injured, okay, and this at the same exact time the patient died, okay, the human brain will appear normal on autopsy because of the immediate death after the injury, okay, as it requires 12 hours after injury for the changes to be evident on H&E staining, okay. So because of the morphological changes we speak about before, the red neurons and the cytoplasmic eosinophilia and all of that, okay, and the spheroids, axonal spheroids, all of these changes are vital processes. So after the injury, if there is no blood supply, if there is no if there is no blood supply, if the patient is not alive, they will not happen. So the brain will appear normal, okay. So we need almost 12 hours after injury for these changes to be evident and can be viewed clearly in the microscopic picture. We're done for today? No, we're not. Okay. No, we're actually done. This is a fast revision. So the first picture is an acute hypoxic ischemic injury in the cerebral cortex. The cell bodies, as we talked about, are shrunken and eosinophilic. They are red neurons, and the nuclei are pycnotic. Number 2, the nasal substance is lost after neural injury. Number 3, central chromatolysis. Central chromatolysis is a histopathologic change seen in the cell body of a neuron, where the chromatin and cell nucleus are pushed to the cell periphery in response to external energy. Thank you so much for listening, and we're done for today.